it is my pleasure to introduce Professor Marty Visscher. She is a professor of skin science. That's sort of the summary at the University of Cincinnati in America. And her research about skin development, adaptation, preterm skin is and one of my favorite topics to read. She writes very well about how we can have a better understanding. She's also very much involved in a lot of our latest skin-related trials, the role of humidity, the coconut trials. And I asked her just to give us an overview of her work and what the current ideas and developments of the skin are. Marty, over to you.
Thank you very much, Kurt, for that introduction. And it's absolutely my pleasure to be talking with you today. I am going to, the session's being recorded. I will make the slides available. And I would be happy to have any questions after we're finished talking, as well as things you might think of after. I have my email address available, and I'd be happy to follow up with any questions. So the objectives today, I have three. One is to talk about neonatal skin development and maturation, kind of what happens. And you see it happening. So this is going to be familiar in kind. The second one is, what can we do? What strategies have we found to help prevent injury and promote maturation? And the third one is to consider the Tiny Baby Collaborative and that work now with a focus on peri-viable and what effect the environment might have on them. But there's a recurring theme throughout my talk. I hope it becomes apparent that a key thing that we're doing with babies and baby development and baby skin development is focusing on hydration. How much moisture is there in the upper part of the skin? So I'm trying to thread that as a theme throughout this talk. So in terms of what do we, I always talk about skin as in a cartoon format because I find it easier to have a visual in my mind so that I understand what's happening. So the outermost layer that faces the environment is the stratum corneum, and its vital functions to provide a physical barrier to protect the human or animal, but in this case human. It provides an acid mantle. It provides a mechanism for tactile discrimination, which is important for high functioning beings. The next part below this stratum corneum is the viable epidermis, and its functions are multiple. One of them is to continually generate new stratum corneum. It also has Langerhans cells, which are part of the immune system, as melanocytes that produce melanin, which is key in skin coloration as well as UV protection. And it continually replaces itself. One of the things that's unique about the skin is that it is a self-renewing organ. Other organs are somewhat renewing, but the skin is designed to be self-renewing, and it's purposeful in that regard. So in some ways, the skin may not get as much attention because it continually fixes and repairs against when damage happens. And then the lowest layer closest to the internal body is the dermis, which provides thermoregulation, mechanical resiliency. So it's all of these layers and how they work together that we're interested in. In terms of the skin microbiome, the skin has a very large surface area on which microbes can interact, and that's because we have the hair follicles that are into the epidermis. And so if you could stretch out and measure the surface area of all the hair follicles and the surface, you would have a very large surface for microorganisms. And microorganisms colonize the surface and in the hair follicles and eccrine glands and sebaceous glands. But they are important for normal homeostasis because they help make antimicrobial proteins, which the skin needs to serve the function of protection and immune function. The microbes will also inhibit pathogenic bacteria, and these microorganisms work with the keratinocytes, which are the viable cells, to create immune mediators. So there's a very much necessity for the appropriate microbiome, and it works on behalf of the organism. Another concept that's important is the concept of what we call the acid mantle or the acid skin surface pH. And just for a refresher, if we show the pH scale as 7 being neutral, where there's equal concentration of hydrogen ion and hydroxide ion, typically water is neutral, except if it flows over rock formations and it'll have other things. But so pH neutral is 7. The skin wants to be in the acid mantle range, which is about 4.5 to 5.5. So that is where all of the enzymes and things function optimally. If the pH, skin surface pH, moves up to neutral and in the alkaline range, then the normal homeostasis is disrupted. So we'll refer to this a bit as we talk. So the enzymes from the stratum corneum that are in the stratum corneum synthesize lipids, and that forms a lipid bilayer structure. The acid pH is necessary for the appropriate desquamation or leaving of the top layer of skin on every day. It's also necessary for inhibition of pathogenic bacteria, and an acid mantle is necessary for the appropriate cohesion among the cells and between the cells, and that provides resistance to mechanical damage. If you take a skin sample of skin and remove the epidermis, the top layer, the stratum corneum, is very tough and very difficult to penetrate because it's an exquisitely interconnected structure. So we want to achieve an acid skin surface pH when possible. Now, so that's kind of just a quick view of the skin. And I talked about it in a very generic sense, but now I want to talk about what happens from birth on and going forward, because there are dramatic changes that happen both in utero during fetal development as well as at birth. So we start off, birth is a colossal transformation. Babies in utero, it's kind of cushy and warm and not so noisy. And then all of a sudden there's this dramatic change to a gaseous environment with a lot of very big human beings talking and carrying on, and they want the baby to cry, so that happens. And so, but the question is, the skin, it's a little wet here in this picture, but the skin is in really good condition. And how does this happen? Because if we sat in water and urine for 9 months, we wouldn't have very good skin. In fact, the top layer would come off like you would have a burn sloughing off. So how does this happen? And what are the clues we have for understanding what has what's been created to allow this to happen. So if we look at fetal development, a lot of work was done by Karen Holbrook and her colleagues in the 70s through the 2000s. And we're going back to some of that now when we have much more premature babies. But if we look at week 4, fetal week 4, we have a basal layer of the epidermis, and then it's covered with periderm. Between 5 and 8 weeks, we start seeing melanocytes appearing in the basal layer. Then from 8 to 14, keratinocytes start being produced and form this spinous layer. And then from 16 to 26, we have a granular layer, and a few stratum corneum layers may have formed. Now, in the periviable infant, as defined as 22 to 23 weeks gestation, is right in this window. So we are now in this part of the biology more than ever before. But as gestation progresses, and we are interested in exactly when this all starts, the hair follicles contain stem cells, and we think it's under the control of the maternal pituitary. These stem cells mix with lipid materials, hydrophobic materials from the sebaceous glands, and they co-extrude along the hair follicle. And we start seeing evidence right around the hair follicle that the skin is changing. It's forming the stratum corneum under there. But eventually, over a period of a couple of weeks, it covers the infant. So eventually we can't detect the fetal heartbeat from a stethoscope on the outside of the abdomen. It's such a thick layer. And when we observe preterm babies, we see a lot of it just before 38 weeks. And what we believe happens is the stem cells are covered with this lipid material, and the material itself is hydrophobic. So we believe it provides a waterproofing layer over the skin so that now it's drier, and we believe that can allow for cornification. Because we know in culture, if we're growing skin in culture, we cannot get a stratum corneum until we lift it to the air interface. So there's something about that lifting, something about that protection from high water that we need in order to form the stratum corneum. So then when the lungs are ready for air breathing, they secrete phospholipid surfactants. And we know from laboratory studies that some of the vernix is detached by that influence of phospholipid surfactants. We can see it in the amniotic fluid. That's how they know the lungs are mature, because when they draw amniotic fluid, it's cloudy, and the cloudy is due to the vernix. Well, then we know the infant swallows amniotic fluid, and we know that the vernix gets into the intestine because people have done studies and find that there's some branch chain fatty acids that are unique to vernix that don't come from any other source. So we know that the vernix is playing a role in getting the intestines ready for feeding after birth. So there's are things vernix does, and again, it makes us wonder what happens when babies are born so prematurely that they never ever have the experience of having their skin develop under vernix. Okay, so now the full-term baby is born And what we know about their skin is it has very low transepidermal water loss. And so transepidermal water loss is the rate of water moving from the epidermis through the stratum corneum. It's the water of respiration that comes through. So if the value is low, it means that the rate isn't particularly high, and therefore we take that to mean there are multiple layers of stratum corneum, and other experiments have confirmed that. So in terms of a barrier, in terms of something covering the epidermis, the full-term infant has a competent barrier. What we then see, though, is that within the first day, the skin becomes dry. And by that, I mean it is dry to an instrumental measurement of hydration. It may also become dry and scaly within the first day, but it's usually a few days after. And so it becomes very low. And then over the first two weeks, it increases. And by about 1 month, it can be even higher than the adult. In this case, we measured mothers in this study. So we've got this tremendous change in hydration. And that makes sense because the baby didn't need hydration in utero. There was plenty of moisture around. But what we see, and you see this all the time, and people ask about it, and parents ask about it, you see this scaliness, lifting scales that happen, and it's not pathologic. It's not a problem per se. It's due to the fact that the skin hasn't had to make anything to bind moisture. We need a certain amount of moisture in order for our skin to be flexible, but it didn't need to do that in utero because it had plenty of water. So we see this dry, scaly. I mean, we did this experiment. We thought, oh, infant skin, newborn skin is absolutely perfect. And then we look at this and say, well, that's scaly. What is that? What's happening? So we figured out what was causing it, and it's because of the biology didn't need to make anything to bind water. So now, okay, so that's a full term. The full term skin also continues to change for a long time after birth. I'll talk more about that. The acid mantle starts forming, but a premature infant is in a difference, and we find it easy. I find it easier to understand preterms if I understand full terms. I find it easier to understand full terms if I understand the adult skin. And of course, we know more about the adult because it's easier to do anything with adult subjects. And the skincare industry, as an industry, has done a lot of research about adult skin. But a premature infant, like the one I show in this picture, this is, I think, a 23-week baby. The skin it's shiny. You can almost see through it. It may have one or two stratum corneum layers at that point in time. This is a photograph, and it looks shiny, and that's because it's wet. You get the specular reflectance from the flash because the skin is wet. And it's with this that you have to try to function, right? I mean, you have to take care of this skin that's wet and very, very fragile. You can see the inflammation and so on. So the epidermis is thin also, in addition to being little stratum corneum. The epidermis on infants is thinner than adults. And the integrity, the transepidermal water loss varies tremendously with gestational age. So this is a graph was published in 1983. And if in this case we have 25 weeks, the transepidermal water loss rate is 75. And if you measure transepidermal water loss over a pan of water, you get about that, maybe a little bit higher. So it's very little protection from the water loss. And you can see it dramatically decreases with gestational age. But now we have babies under 24 weeks, so they have even higher values. So now we're dealing with this type of skin more than ever before. Even for a 25-weeker, even 1 month later, 4 months old, or 4 weeks of age, their transepidermal water loss is still higher. It's in the 20s, still higher compared to a full-term infant. And if you look at the dermal structure by histology, this is a cartoon of the full term. It has this dermal structure, but it changes a lot. In a full term, we did some work on a little bit older babies and showed how the elasticity and viscoelasticity really change in the first couple of months. But a preterm infant has less fewer connections to the epidermis, fewer components to the dermis. And so it means it's not resilient to anything that happens, such as rubbing, or if the infant moves, the infant can remove the top part of the skin just from movement because it doesn't have the structure. So this all has to develop too. So I want to talk a little bit about skin hydration and why is it important? Because if you look at the skin, and this is for anybody, but in an adult skin, in an older infant, if you magnify the layers here, you see that these cells have filaggrin. It means filament aggregating protein. So filaggrin aggregates the keratins and holds them together. Because again, it's trying to produce a very tough structure. Eventually, as the cells move up closer to the environment, this filaggrin undergoes degradation, proteolysis, to form small molecules, and they're collectively called natural moisturizing factor. They're small amino acids and the degradation products of those amino acids. And so when you have this production, the filaggrin producing the natural moisturizing factor, shown here by these small dots, allows moisture to be bound to the skin. And so ideally, you have not too much moisture, because that'll cause the skin to be damaged, but not too little moisture, because then the skin won't function. The question is, what's the ideal moisture? And the answer is, I can only guess, it's probably a reading of 130 on an instrument, but that's not very helpful. It's just that we all have to have that level of hydration in order to have the skin function and remember, it's going to be self-renewing. So if you look at the process of self-renewing, and we'll look at it just through this animation, we're losing the top layer about every day, unless there's something wrong in which we don't lose it and it gets very dry and scaly, and we don't lose it because it doesn't have enough moisture in it, or we lose too much of it because it's damaged in some way. But normally it's supposed to be 1 layer per day, and it gets replaced from underneath. That's a good thing for the preterm infant, for the periviable infant, to the extent that the infant has the machinery to repair itself. Okay, now I want to just touch on the work we've done on what happens over time. And the time frame I'm talking is a few months after birth. What is happening to this skin? We know you see the changes in the clinic and you talk about them and describe them. That's how I know about them, because when I only know anything because I've been around nurses and clinicians and physicians and therapists to be able to explain what they, or see what they, learn what they see. We can today, in today's world, take a small piece of plastic, it's a disc, about 30 millimeters. We can put it on the skin. If you can do this experiment with any piece of clear tape, put it on the skin for a while, and we usually leave it on for a minute, and then we take it off. And when we remove it, we see these white flakes. This is actually the stratum corneum layers that we're getting ready to come off. Now, we can take these materials, and through the omics technology, proteomics technology, we can analyze a variety of proteins that are ultimately byproducts of the products of gene expression. But we can analyze things that are in the stratum corneum. That helps us understand what the state of physiology is. So we did some experiments, and we've published some of them, where we did the biomarkers from this. We can get maybe 3 sequential before the baby gets upset. And if the baby's upset after 1, we don't take more than 1. But we can do this. Now, it's also true that when you put something on a preterm and it comes off because of hydration or because of humidity, that whatever it is, that tape or the electrode has this stratum corneum on it, and it can be used to analyze, to figure out what's going on. That's what's very exciting about all of this now, because we're accidentally getting it. We don't have to go in and take some more because we accidentally get it when things come off. But anyway, so that's where we do proteomics. And then we did another part of the study where we were able to get full thickness tissue at the time of surgery. The babies needed to have a surgical procedure. So when they put a laparoscope in, they would make an incision, then they would give us a full thickness piece that we could snap freeze and then analyze later. So what do we learn from proteomics? What we learned was I'm showing you the results, levels of antimicrobial proteins from the tapes at 2 time points. This is a few days after birth, and this is 2 to 3 months later. So because we were collecting these samples sequentially or longitudinally. And what we found was these antimicrobial proteins are as high as the levels in adults, except in preterms, this S100A7 was lower. But 2 to 3 months later, these babies are producing antimicrobial proteins at levels way higher than adults. So they are producing antimicrobial proteins. And we're working now on what's the rate of change look like. When does it ramp up? What's the rate it happens? So the biology is programmed to make these antimicrobial proteins. The other thing we see, and these are the abbreviations, serpines and aliphin. These are protease inhibitors, enzyme regulators. And you can see that with a few exceptions in the preterm, the very preterm, these are still higher than adults. And why is that? It's because we don't necessarily want these proteases to be removing the skin that the baby just built. So we want some of these things not to go as fast. And again, 2 to 3 months later, the levels were still higher. So I've just highlighted a few. We're putting the picture. Why are these so important to the infant? What do they do to the biology? Why are they there? What are they doing to the microbiome? That's another question. And then finally, the last group I'll talk about are filaggrin. These are the proteins, various proteins that are involved with processing the filaggrin, helping it produce natural moisturizing factor. And again, these are high levels at birth, and they're still higher than adults 2 to 3 months later. But it's amazing that preterm infants have less of it than the other late preterms and full terms, but they still have higher values than adults. So they are ramping up, and by 2 to 3 months after birth, all of these preterm, all of the neonates have higher levels of the natural moisturizing factor than adults do, than their parents. So again, it's an important aspect, and these materials at NMF are in large part responsible for producing the acid mantle. So the idea here is to connect the biology with what the infant needs in order to combat infection, etc. And then I'll just briefly, in the transcriptomics, we compared, so far we published on newborn infants versus adults, and we see that there were 143 processes increased. The most significant were extracellular matrix organization and also structural organization. Whereas in adults, there were 101 processes increased, but they were involved with epidermal development, maintaining that homeostasis, maintaining self-renewing, keratinization, cornification, and some of the adaptive immunity. So the babies, the young ones are involved with this extracellular matrix, and the question is why? This is what it looks like in the map. That's not so important, but extracellular matrix genes regulate proliferation, differentiation, all of these properties. They are it's continually modified. It's involved in wound repair, infection control, and so the infant is programmed to produce it. Now we've getting ready to publish a paper on premature versus full term. Again, they're very different. The prematures are involved in metabolism more, and the older full terms are more involved with producing the barrier. So again, I think we think this is going to help us understand what we can then do but again, we can't wait necessarily for the science. So I want to transition into what can we do? How do we think about preventing this injury and promoting maturation? And this is related to the periviable, because the periviable is top of mind right now with a lot of effort going on, particularly hats off to Pranav Yani and his colleague and folks in Australia. You're really leading the charge in some very exciting science here. But what we've done is gone back to some of the work that Holbrook did, and we see that even at 20 weeks, there's a little bit of scale cornification that happens, and this is before we are covered with vernix. So why this happens, and the fact that it does happen means that perhaps the babies are beginning the cornification process, and maybe we can take advantage of it in the care processes. A little bit older, at 20 weeks, we see more stratum corneum. So going back to this, we're also looking at a range of gestational ages, revisiting histology to see if we can understand where the vernix production becomes most active. Okay, so what we want to do is think about transepidermal water loss. And transepidermal water loss is inversely related to humidity. If you're a premature infant on the right. So the higher the humidity, the lower the transepidermal water loss. And because they lose fluid, because they have a poor barrier, we have to put them in, we have to do something to keep the humidity in. And so, as you know, what's being done is put them in high humidity, or in the case of Iowa, they wrap them in saran wrap and put them under a radiant warmer. We don't know what their skin looks like I don't know what their skin looks like. I haven't been to Iowa to look at it yet, but they probably are creating humidity right next to the skin surface. And it will be intriguing to see what the differences are in those environments. But in any case, the effect of humidity is pronounced in premature infants. In full-term infants, it really doesn't affect it because they have a good barrier. So adding more humidity doesn't markedly change transepidermal water loss. Okay, now I want to talk about the role of the incubator humidity because we talked about hydration. And so here are the levels of care. And I'm borrowing from Dr. Yanni's study and Matt Raisavi's study, and we'll talk a little bit more about this, looking at varying the 40% in Iowa, but that contrasts to other people who are usually above 70, and it can be 70, 85, 90, 95, as much as 100%. So what happens is the humidity keeps the water in. And so the incubator is keeping the water and preventing dehydration. So its job is to keep the fluid in. So the question is, what is that doing to the skin? Because people want to put things on the skin to be able to read vital signs, etc. And so we have to keep them in high humidity. We have to preserve their fluids. And yet we still have their skin needs to develop. So that's the trade-off of how do we manage? It's all about managing hydration at this point in terms of caring for these infants. And it's very difficult. So I'm sorry, let me go back. Okay, so I went back to some data that our group, my predecessor, the late Stephen Hoth and his colleagues generated. This was a study where we had babies, some babies less than 30 weeks, some less than 27 weeks. And then we had some 32 to 34, and then a group of full term in the NICU. And so we measured a parameter called hydration, and it's measured by a very simple instrument that uses capacitive reactance, and it's measuring the electrical conductivity basically of the moisture on the surface. But what we found, regardless of humidity levels, we found a very high hydration, in much higher hydration for the younger than 30 weeks here, and in this case, younger than 27 weeks, compared to the full terms and older babies. So was a big difference in hydration levels. And then we followed them, the youngest ones, the ones in humidity, followed them for 5 days. So day 1, you have a very high hydration, but by day 5, it was lower. And it was almost in the range of what a full term would be. So something happens over 5 days, even at 70% humidity. And the question is, how does that happen? What is the humidity? Do we actually have lower humidity because we're opening and closing, or what's really going on? So those are some of the questions that we're going to answer right now. But we know that the skin barrier is going to be less permeable after a few days, according to this initial data. So this is something we need to understand further. So how do these two things, how do varying humidity levels and gestation affect skin surface hydration? How does it affect the way you have to handle the babies? Well, usually we put all kinds of things on them, try to, for various purposes. And if the skin is wet, they won't stay. They won't stay on the skin. If the skin is wet, it won't cornify as well as it would if it had some drying. So what happens is this wet skin in these environments to maintain fluid will become more permeable. If you put something on, the skin's more permeable. It also has a higher friction. So if the baby moves on the blanket, it's going to cause an injury. And then you have less adhesives. So what happens is, because things don't stick, we tend to put more aggressive adhesives, and that causes more damage. So this is a situation that's very, very difficult to manage, and it's the challenge is what do we do? How can we optimize the process of caring for the infant without causing injury? So just what happens is these occlusive things like electrodes or certain tapes will block the evaporation that is occurring, even though humidity is going to keep it low. So, and then when that happens, it's more permeable. That's all I'm showing here. And it to irritants. So we know from experience. I mean, these are pictures from our unit where we've caused injury. One adhesive tape product can remove most of the epidermis. And it has happened before, not intentionally, but it's happened. So figuring out what to put on, how to keep it on long enough. Okay, so the skin's hydrated. You have to look at the anatomy. In the neck folds, the axilla, behind the knees, where the ankles are flexed, the wrists are flexed, in between fingers, any place where you can have skin against skin, they're going to accumulate moisture, and that's going to cause problems. Temperature probes, central lines, anything. So, and then of course, then we have the NIV and CPAP masks. Bedding. Some of the bedding is washed in hot water. It's woven. It's got a thicker, maybe a flannel kind of weave. And if you look at it under a microscope, it's really bumpy, and it's got all kinds of cotton coming out of it, and that's helping it stick to the infant. So even the bedding, even using a flat cotton that might absorb some of the moisture, is going to be better than using something that's rough, that's been through a washing process. So trying to manage moisture, and in doing this, we try to create drying when we can. So we put something in the folds to help pull the water away so it won't be on the skin surface. And so I'm going to show you a series of things that we found, and this was we tried different things. We had to see what worked and what didn't work, and we learned, look, the kind of injury we can get. This baby was 9 days old, and we still had too much moisture. We see some cornification, but we still within the folded areas is what we have to be worried about. So what we do to try to manage hydration, we use a product, the name of the product, it's a brand name made by Monlecki. It's called Mepilex Light. It's a foam. It has dimension to it. It has a silicone adhesive, which doesn't cause damage. It also doesn't stick very well. So it's going to come off and have to be put on, but it will help pull moisture into it. So putting it in to protect the folded area, putting it around a neck can help manage that moisture. And we've learned by trial and error that this one is an effective one. We'd like to have a way to pull that moisture away at a rate so that we can not take fluid away from the infant, but take that surface moisture so that the surface can cornify. So it becomes a plan of care of just diligence at everything you do with this baby. It's not perfect. We're still learning. We have a lot to learn, and everybody's trying to learn together. The dressing might have to be replaced every so often. We found this because we had one that wasn't working very well. So this purpose of this is it shows even after day 6. At day 6, the skin looks more cornified. It looks drier, and if you look at it, magnify it, you'll see the detail of the surface, which is telling us it's cornifying. This is an example where we learned the hard way. We were using a dressing called Mepitel 1. It has a thickness to it, but it has big holes. It's designed to be put on the skin, and then on top of it, a secondary dressing is designed for wound healing. We were using it to try to protect the skin. And what we found was, in this picture, you can see that actually the skin was poking through it, because the skin is going to be developing. But we saw that the skin was just stayed wet. It wasn't, because didn't have a way to pull any moisture away. It just accumulated under the areas that didn't have holes. So, but this, just really seemed to make it worse. So we tried it, didn't like it, and then tried experimenting. We had to experiment with babies with different dressings, putting them on a different one way with adhesive side down or with not adhesive side down, and just observing and seeing when it started looking better. So that's how we arrived at this. And again, it's one of those things where it would be lovely if some of the companies who make these dressings would kindly work with us so that we could have a dressing that would do what we needed to do. And that's another part of my soapbox is working with partners like that who can help reduce some of these ideas to something practical. And again, just if you see it, you can get all kinds of levels of loss. We've been really trying to minimize the use of any topical agents like chlorhexidine, only putting them on areas where we absolutely need them to be. We would cleanse an infant with sterile water, blot dry, and then cover it with the dressings, the non-adherent dressings. Sometimes we can take a big piece and then hold it in place with a Covidin or another type of tape that I'll talk about in a minute. This isn't it isn't the best picture, but we started putting nothing on the skin with the umbilical lines. They put them in, they leave a long tail, and they do the suturing, and then they encase the end of the line with clear tape and put a cotton tape on the outside so there are no rough edges. And this just sort of slaps around. But we try not to put any more adhesive on the abdomen because we see so many wounds, and some of it's from putting a lot of chlorhexidine or betadine, and that has happened to pool on the skin. So trying to be very judicious about how things are done. It's a bit of a culture change, but people are seeing the benefit. So we're going to be creating a series of videos on how to do this as soon as I can wrangle the nurse practitioner into making videos for us so that we can have them available to show people how to do it if they don't. Again, trying to do everything we can to minimize skin contact. Things that we used to do just always we'd swab the abdomen, but these babies it's a different story. Another thing we do for probe covers, the reflective probe covers, is use something called Mepitac tape. It's again made by Monlecki. It's not particularly adhesive, but we cut out a area so the probe can be against the skin, but then the cover isn't against the skin. The cover is against the probe and then against this tape, so that we're preventing some of the damage that can happen from these probe covers. We in our unit do not use adhesive remover of any kind. We use water and a lot of patience. I know the nurses are interested in trying other things, but we haven't done that yet because we want to when we do anything like that, we want to be documenting what happens. Here's some examples of the pressure injury, the injuries we see that relate to CPAP or NIV. You can see those. We have a biomedical engineering department is working on, we've got a sensor now that can measure the pressure between the mask and the skin. We've done it on a mannequin and that paper's coming out. Next step is to do this work on babies to see because the pressures we measured with this particular sensor are very high, and we want to really understand that biology. We rotate between mask sizes if we can and still get a seal. And then we rotate between a mask and prongs. The babies do not like prongs. They don't like them any time of day. Doesn't matter. They don't like them. And so is there something we can do from an engineering standpoint to avoid those injuries? We're looking at it. But this is something that's getting some attention. It needs more attention. Certainly CPAP is better. So we use something between, it's this, you can see it here, it's duoderm. We stick it to the prongs so that we provide some cushioning, and the prongs don't go up as high into the nose and cause irritation. Okay, and so Kurt asked me, does environment matter? And the question is, I wish we knew. We wish we knew. We wish we could take the data we have and really understand it. But we don't. So your colleagues in Australia are embarking on a really nice trial to look at two humidities, 80 and 95%. And there's a group in Texas with Matt Rysavi who's looking at 70% and 90%. And they're doing sub-studies. I mean, the main study is to look at fluid management and sodium levels to try to answer the question of what is the effect of humidity. And then there are some sub-studies on smaller portions that are going to help understand what the skin is doing during the time of 14 to 28 days. So it's very exciting because they've been very gracious about sharing everything and inviting us to participate. I'm very impressed with all the work that's being done and the diligence. So I am very confident that a lot of learning is about to happen. The question is, does it make sense? Does it matter? We've looked at this is just a summary slide I made of some of the historic data that goes back, and in 1982 and 83 they weren't using much more than 50 or 60% humidity. That's a fairly new thing. And then Johan Agren is the one who's done a lot of work with babies at 80 and 85% humidity. The problem is when you measure transepidermal water loss, the measurement is affected by humidity. It's so you're going to have to take the infant out of humidity, which nobody really is too keen on, or you have to correct the humidity to 50%. And I think we'll have data that under these conditions from a lot of situations, and we will understand. So I probably have gone over time, but thank you for your attention. I'm happy to answer questions.
This was wonderful, Marty. The overview and academic sort of background about skin development, skin function. You highlighted some of the aspects, particularly I found really interesting and new to me about the extracellular matrix, where I'm assuming most of the water is actually located, but how that still has to form in these little babies. Then you mentioned about transepidermal water loss, and how there are tools to measure, and different scenarios can be described where the older data might not be as applicable. So we need this new data. And then you showed these wonderful examples of, how to manage sometimes, like you said, by trial and error, finding out which product is best suited for certain types of skins. I'm going to go, there's some questions already posted. One is from Jenny. She asked about bathing of preterm infants. Is that good? Is it bad? Do you need soap or not?
We have had tremendous discussions about that. Some of them quite heated, actually, because people don't want to leave any contaminants from birth, especially if they're choreo babies. And so what we are comfortable with is using, and again, we're talking about 22 and 23 weeks babies, using water, that's at the temperature of the incubator, and taking many different care times to remove some of those birth contaminants. Being very, don't rush, but being very deliberate and doing it over multiple cares to remove some of those things. The problem with what do we bathe them with, and that's the other part of the background I have, is in skin care formulations and skin product interactions. And there are very few things that we would want to put on a baby because there are too many opportunities for exposing them to allergens. We found a few cleansers that have ingredients that are mild, but the question is, does it really help? And the literature doesn't give compelling reason to use soap and water. Now, that said, parents and nurses want to do this bathing. It's a process that's important to the normalcy of these infants. So we can't ignore it forever. We need to come up with a better idea. And I think doing it with water can be okay. We like them to be 28 weeks corrected before we use a cleansing product on them. We, in our unit, do bathing twice a week. I don't know what it is in other places. I think it varies a bit. But the data on, see, the thing is, if you expose them to water, you're going to remove the natural moisturizing factor they've been working hard to make. So, and because we know that from the experiments we did. So that's the best we know.
There's always a balance to be found between what's indeed practice in different countries, even religious sort of
Right.
backgrounds, and actual, what will be
Right.
scientifically best. You mentioned a lot of products have silicone gel. Louise has a question about Strataderm. Is that a product you're aware of or using?
I am not, but I will look it up and get back to you. Strataderm. I will look up that product and give you an overview of how I would, because that's what happens. We have to look at it, look at the ingredients, and make some some judgments, but I'll be happy to do that.
Oh, that'd be excellent.
See, and that's the other thing is the companies are not necessarily very transparent in what's in there. They put some of the information on the labels, but it's like, tell us more, show us the data. So
yeah, I bet you're in a good position to get that information from some of the companies, so that is always sort of helpful. Sam asked a question about the effect of phototherapy particularly in these tiny babies, in the pre-viable ones.
So that's another very, very good question. I wish I knew what it does, because it's blue light therapy. Does it facilitate stratum corneum development? I have no idea, and I don't know how we would do an experiment to find out exactly. I mean, we'd have to have I guess the diaper area there, if that's not exposed to phototherapy, but most of the rest of the skin is. So
yeah. And we treating more and more very early bilirubin values just because we get them. We don't ask for them, but they come on the point of care, they reach a certain threshold, right, and there seems to be more treatment very early on. So that can be impactful. All right.
Right.
It is 1:02. Marty, I want to thank you so much for this excellent overview and talk. There will be more questions. I'll probably will ask you some of the questions, but Marty's email is on here. Feel free to ask her directly as well. Thank you all for attending. Based on this talk, I will certainly ask Pranav to present here as well on his trial and the ideas sort of behind it. And next week, there is no session, and we will be back on the 17th of October with George Smolzer from Canada talking about what's new in resuscitation. All right, Marty, thank you so much. And
you are most welcome.
Thank you.